My friends, every day I cross paths with friends and acquaintances and complete strangers who are agnostics and atheists, and they say to me, Eden, we don't believe in God, and we find it absolutely impossible to believe in the notion of a creator when there is no tangible evidence, and in a world that is enveloped by anarchy and chaos and shameless bloodshed. We can't possibly believe in this notion. We can't. And they say to me, if you can prove it to me, if you can show me tangible evidence of God, then we will reconsider. So I wanted to take a second here and show you what I consider, and any rational, reasonable mind would have to consider as tangible evidence of a higher intelligence and a grand designer. I want to take a few seconds here. And I actually want to take you on a short tour of the human cell. Just in case you haven't taken a look recently, let's take a look right now at the human cell.
It's like going into an automobile factory. The factory has a large number of machines. The parts have to fit together in very specific ways to do their jobs, and if things go wrong, the cell is in big trouble. And just one cell is enormously complex, but humans, you and I, are made from trillions of cells, and those trillions of cells have to fit together in the right way and do their own job. Darwinism was a lot more plausible when we were thinking about globs of protoplasm than it is when we're thinking about molecular machines.
Each of these biochemical machines is a masterpiece of engineering and nanotechnology. They are essential to functions as vital and diverse as vision, photosynthesis, and the production of energy in the cell. Michael Behe has studied several of these machines, including the flagellum, a remarkable rotary motor.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It had a propeller and the hook region and the drive shaft and the motor. And I looked at that and I said, that's an outboard motor. That's designed, you know, that's no chance assemblage of parts.
Behe's reaction was not surprising, especially when the bacterial flagellar motor is animated and magnified more than 50,000 times to display the details of its construction and operation.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
It's got some tail proteins which act as the propeller. When the flagellum rotates, these push against the water and therefore push the bacterium forward. And the motor uses a flow of acid from outside of the cell to the inside of the cell to power the turning.
The bacterial flagellum has 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller. It's not convenient that we give them these names. That's truly their function.
In all, about 40 different protein parts are required to build a flagellar motor. Half of them are constructor proteins, specialized mechanisms that assemble the flagellum's individual components. Since its discovery, biologists have tried to understand how a machine of such superb design could have arisen gradually, without foresight or plan. In his study of evolution and molecular machines, Michael Behe has raised a significant challenge to the creative power of Darwin's mechanism of natural selection. It is called irreducible complexity.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
Irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function. Catching mice. The concept of irreducible complexity also applies to biological machines, including the bacterial flagellum.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell. You can't put something like that together gradually because they need a large number of parts interacting with each other at the same time before they work at all.
Without the tools to observe the machinery of the cell, and long before the idea of irreducible complexity, Charles Darwin offered a way to test his own theory. In Origin of Species, he wrote, "If it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down."
Neo-Darwinism and its associated theories of chemical evolution and the like will not be able to survive the biology of the information age, the biology of the 21st century.
Meyer's conclusions are based upon his understanding of the DNA molecule and the genetic instructions that are locked within the nucleus of living cells.
In 1953, when Watson and Crick elucidated the structure of the DNA molecule, they discovered that DNA was a carrier of genetic information in the form of a 4-character digital code. That is to say that DNA functions like a software program, only more complex than anyone has ever created or devised. For a biological system to run and operate, it needs genetic information to build the proteins and protein machines that cause the cells to maintain their function.
This information is stored in a precise arrangement of 4 chemicals that scientists represent with the letters A, C, T, and G. Sequences of these chemicals provide the instructions necessary to assemble complex protein molecules that in turn help form structures as diverse as eyes, legs, wings, and hearts. This code has been called the language of life, and it is the most densely packed and elaborately detailed assembly of information in the known universe.
Well, lots of people have wanted to explain the origin of information by reference to the laws of physics and chemistry or by reference to the chemical properties of the constituent parts of the DNA. But that would be like saying that you could explain the information in this morning's New York Times headline by reference to the physics and chemistry of ink bonding to paper. There is a chemical explanation as to why the ink sticks to the paper, but that does not explain the way the ink got arranged to convey a message that could be understood by speakers of the English language. Information requires a material medium, but it transcends the material medium.
An explanation for the origin of the genetic instructions needed to build the first life is the holy grail of 21st century biology. Theories proposing that this information arose through natural selection acting upon non-living molecules, or the self-organizing power of chemicals in a primordial soup, have repeatedly failed. Even time and blind chance, the oft-invoked saviors of implausible biological scenarios, have fallen far short as accounts for the source of the instructions in DNA. Mathematicians, for example, have calculated that a universe filled with monkeys, typing relentlessly throughout the oldest estimated age of the cosmos, would have no realistic chance of producing Shakespeare's play Hamlet. let alone a transcript of the genetic information required to build even the simplest living cell.
Based on our uniform and repeated experience, which is the basis of all scientific reasoning about the past, there is only one known cause for the origin of information, and that cause is intelligence. Whether we're looking at a hieroglyphic inscription, a section of text in a book, or a computer software, if you have information and you trace it back to its source, invariably you come to an intelligence. Therefore, when you find information inscribed along the backbone of the DNA molecule in the cell, the most rational inference, based on our repeated experience, is that an intelligence of some kind played a role in the origin of that information.
Now, due to the technological advancements of science and the tools and instruments that they use, we can now see exactly what's going on a cellular level inside the human body, and it's absolutely mind-boggling. You could see the molecular machinery working 24/7 with the kind of meticulous precision that our minds can't even conjure, understand. Like it makes a car factory look like a child's play, for God's sakes. It's unbelievable. And better yet, and bigger than this, and more concrete evidence, we see information in the DNA molecule, in the backbone of the DNA molecule. We see information, intricately embedded information into the cells of our body. It's unbelievable. And we know that information presupposes a higher intelligence conclusively. And a higher intelligence presupposes a divine creator, a prime mover. If this is not concrete evidence, if this is not the very signature of God and tangible evidence, I don't know what is.